Hi, I'm Dr. Leslie Blankenship-Williams, and this lecture covers cellular metabolism for human physiology. Now, that's just a fancy way of saying chemical reactions that occur in a human cell, of which there are probably hundreds, if not maybe even thousands. Of course, we're not going to cover all of them. We are going to cover the chemical reactions and what is known as the metabolic pathways that specifically deal with this question. How do cells pay for work? Before embarking on this video, please make sure that you have completed Chapter 10 of your workbook. This lecture assumes that you have already completed that chapter, and if you have not, a lot of this is probably going to be over your head. So for your sake and maximizing your learning without having to rewatch videos, I strongly suggest that you pause this video and complete Chapter 10 prior to watching this. All right, so let's back up a little bit and ask about what type of work would a cell conduct. So by definition, work is something that requires an energy payment into it. So there's actually three different categories of actions that a cell takes that is defined as work, meaning that you need to have some sort of energy input into that system in order for it to occur. Now, one of which is non-spontaneous chemical reactions. From Chapter 10 of the workbook, you've learned that this means that the products have more chemical energy in them than the reactants do. Now, the cell needs to be able to do a variety of non-spontaneous chemical reactions, but those chemical reactions require some sort of energy input. They are not free. So, therefore, this would be a type of work. Now, another category is movement. And this can be movement that you could potentially see under a microscope, like a flagella moving in a sperm. Or it could be something like the vesicles moving along the monorail tract of the cytoskeleton. Or it could be something like a muscle contraction. Those are all types of movement. And then the third category is active transport. Now, by definition, active transport is any transport that requires an energy payment. So typically, what we are referring to is creating a concentration gradient of a substance. But it can also refer to bulk transport, like endocytosis and exocytosis, both of which require movement and therefore require energy payments. So now that we've covered three types of work that a cell might do, let's get to our real question, which is how does a cell pay for this work? Now, from Chapter 10 of the workbook, you know that there are two types of potential energy that a cell might use. By far, the most common is chemical energy, the energy stored up in covalent bonds. But sometimes concentration gradients are used as well. Now, if you recall back to the transport lecture, you can see an example of concentration gradients being used with secondary active transport. We saw this with the sodium glucose cotransporter. We allow sodium to diffuse down its concentration gradient as long as it brings glucose, the unwilling friend, with it into the cell. Okay. So that's a look at concentration gradients, which are sometimes used to pay for work. Most often, chemical energy is used to pay for work. And although chemical energy could be in a variety of different molecules, the molecule that the cell has adopted as its energy currency is going to be ATP. So ATP is a type of nucleic acid that has 3 phosphate groups attached to it, and each phosphate group is negatively charged. So when you put all 3 of them together, they really hate being next to each other, and so that last phosphate group in particular really wants to jump off, and so that represents a type of potential energy, as having that 3rd phosphate group on where it really doesn't want to be. So ATP is our currency. That's not to say that it's our only source of value in a cell. And we're going to assume that the cell is a little bit like a mini economy. If you look around your household, there are lots of things that are probably worth money, but you can't take like your $200 couch to the grocery store and load up your cart with $200 worth of groceries and then ask the, clerk if they'll take the couch as payment for the $200 worth of groceries. And while the couch might actually be worth $200, couches are not an acceptable form of currency in a grocery store. Instead, you have to do a two-step process. You would have to sell your couch for $200 to a willing buyer, then get the cash, then take the US dollar to the grocery store where you could then buy groceries because the US dollar is the accepted currency. In the same way, ATP is the accepted currency. It's like cash or money to our cell's economy. However, there's a lot of biological molecules out there that are certainly worth some ATP energy, and they just need to be converted into ATP in order to spend it. So much of our question is going to be about how do we make ATP from these other molecules like fatty acids or glucose or even amino acids. How can we harness the energy in those to make ATP? Much like how would you sell a couch to get $200? So that's going to be our driving question. Now let's quickly review ATP. So this is ATP. You can see there's the nucleic acid, and then there's the 3-phosphate groups, and since they're all negatively charged, they hate being next to each other. When you spend ATP, so to speak, like spending $1, all that really means is that last phosphate group detaches. So when the last phosphate group pops off ATP, that provides the energy that will pay for work. Now, going from ATP to ADP, adenosine diphosphate, and potentially an additional phosphate group, is really easy. It's just like if I were to say, here's $100, how are you going to spend it? In your head, you have it spent in 5 seconds. It's very easy to spend money. The more difficult question is, how do you make money? So just like real life, the easy question is, how do you spend ATP? The difficult question is, how do you make ATP? So that is going to be the topic for today's lecture. What chemical reactions occur that allow us to add that 3rd phosphate group back onto ADP to make ATP? So again, that's going to be our question. What can we do to reattach that 3rd phosphate group to ADP so that we can regenerate ATP and therefore go spend it on something else? Okay, I'm going to take you to my drawing pad where we are going to start talking about some of those processes. So it's very common for students to get lost in the weeds of all of the chemical reactions and lose sight of the main question. What we're trying to do is phosphorylate ADP to make ATP. So that's what making ATP is. It's attaching that 3rd phosphate group back onto ADP in order to make ATP. Now recall that ATP is a very high energy molecule, whereas ADP with the separated phosphate group has less chemical energy. So technically this is a non-spontaneous chemical reaction. So therefore, in order to run this chemical reaction, we would have to be able to add some sort of energy to the system in order to get the phosphate group back onto ADP. So how do we get that energy? Well, this is where cellular and aerobic respiration comes in. The energy needed to phosphorylate ADP is actually stored up in the non-polar covalent bonds of other biological molecules. And this is harnessed or harvested via the very complicated metabolic pathway known as cellular or aerobic respiration. So the reason why it's so complicated is because we're trying to maximize the amount of ATP that is generated per fuel molecule. And by fuel, I mean one of the biological molecules. So again, we're going to eat glucose, sugars, fatty acids, amino acids, and then we're going to break them apart, capture that energy through a series of complicated chemical reactions, and try to phosphorylate ADP into ATP. So aerobic respiration in a human cell is broken down into a couple different stages. And you've probably learned these stages if you've taken a lower division general bio class, such as Bio 100 or Bio 102. So briefly, they are glycolysis, which happens in the cytoplasm, and then everything else happens in the mitochondria. So we have our second stage, which is called the Krebs cycle or the citric acid cycle. Now, why they pick different names for this, I don't know. But both phrases are used to describe the same set of chemical reactions. I tend to prefer citric acid cycle simply because that's what I've been taught. And then finally we get to a really complicated phrase called oxidative phosphorylation. That's going to be our final stage. And this is going to be where we get our big ATP yield. Now what we're going to do is we're going to briefly cover glycolysis. I'm going to really briefly cover the citric acid cycle. And then I'm going to cover oxidative phosphorylation in some detail because it's super important. Now, once we cover all of those stages again, then we're going to take a look at what happens if we lose our oxygen. So oxygen is required for this process. That's why the term aerobic is in there. That means oxygen. And if we lose our oxygen, it interferes with our ability to make ATP via this very long but efficient pathway. So then what do we do? And we're going to take a look at that too. All right, let's get started. So aerobic respiration is commonly broken down into three stages: glycolysis, the citric acid cycle, and oxidative phosphorylation. Before getting into the details of each stage, let's first take a look at where these stages occur in the cell. Now, most of them occur in the mitochondria. So let's quickly review the mitochondria anatomy. It has two membranes, an inner and an outer membrane. Those two membranes create two compartments. The compartment between the two membranes is known as the intermembrane space, or between two membrane space, whereas the most internal compartment is known as the matrix. Now, glycolysis actually does not happen in the mitochondria. Glycolysis happens in the cytosol. So we start with glucose, which is where the glyco comes from, and then lysis means to break. So we're going to take glucose and through a variety of steps break it down into two molecules known as pyruvate. Then the pyruvate is going to get shuttled into the matrix. And from there, it's going to further be broken down via the citric acid cycle. And then our last stage actually occurs mostly in the inner mitochondrial membrane, and that is oxidative phosphorylation. So now we're ready to learn about the three stages that comprise aerobic respiration. We'll start with glycolysis. Now the name glyco implies glucose. And of course, glucose is by far the most common fuel source, but it's not the only one. Fatty acids, amino acids, and occasionally nucleic acids can also be broken down by a similar but not identical metabolic pathway. So although we're going to learn about how we break down glucose, please note the other fuel sources like fatty acids and amino acids can be broken down using similar pathways. All right, let's get started. We are going to start with glucose. And interestingly enough, glucose is actually pretty stable. So before we do anything to it, we actually have to invest 2 ATP to make it less stable so that it's easier to break apart. So the very first part of glycolysis actually involves spending 2 ATP. So if you want to think about this like an economy, then we would be 2 ATP in the hole. In other words, we've invested 2 ATP before getting anything back. This is what is known as the ATP investment phase. Now, I have only 3 arrows drawn, but it's actually more like 5 steps. And then once we make glucose pretty unstable, it's easy to snap in half. And the molecule that is produced as a byproduct when we snap glucose in half after modifying it somewhat is called glyceraldehyde 3-phosphate, also known as G3P. So once we have our 2 G3P molecules, then it's time to finally get some return on our investment. So the first thing that happens is NAD+, which is our electron shuttle, our taxi cab, or our coenzyme, is going to pick up some electrons and a hydrogen off that molecule and become NADH. So we're going to reduce our NAD+ to NADH. Then we finally get some ATP. So we actually do this in 2 separate steps, but we convert some of the chemical energy directly into ATP. And the molecule that we have at the end of this process is known as pyruvate or pyruvic acid. They mean the same thing, they're just 2 slightly different states of the same molecule. I use them interchangeably. Now the steps that are drawn from G3P to the pyruvate are for 1 G3P molecule going down to 1 pyruvate molecule. But there are actually 2 G3P molecules leading to 2 pyruvate molecules. So technically all of these yields should be doubled. So instead of 1 NADH, we actually have 2. And then instead of 2 ATP, we actually have 4. So let's cover some quick bookkeeping about what we invest and what we get back in glycolysis. So the investment phase invests 2 ATP up front, so we start out -2 ATP. But then in the second part of glycolysis, we actually get some stuff back, including 4 ATP and 2 NADH. So when we add all of this up, our profit is going to be 2 ATP and 2 NADH. So again, that's our profit from glycolysis, 2 ATP and 2 NADH. Now that we've finished with glycolysis, let's take a look at what happens to pyruvate as it proceeds to enter the mitochondria. So remember, our next stage is going to be the citric acid cycle, which occurs in the mitochondrial matrix. And we end glycolysis with pyruvate or pyruvic acid. Now what we want to do is we need to shuttle that into the mitochondrial matrix. Now some textbooks will actually treat this as a separate step, but I'm going to kind of lump it in with the citric acid cycle. So that pyruvate molecule needs to cross both the outer and the inner membrane of the mitochondria to get to the matrix. As pyruvate gets shuttled in, that last carbon with the carboxyl groups attached are going to turn into carbon dioxide. So that last carbon is actually going to get cut off and become carbon dioxide. Now the hydrogen that's there is going to get added onto NAD+. So as pyruvate gets shuttled in, the last carbon is going to get cut off and become carbon dioxide, and that's going to be a waste product. And the hydrogen that's there is going to go on to NAD+ and become NADH. So what's left is what is called an acetyl group. And the acetyl group gets attached to a stabilizer known as coenzyme A. And once we have acetyl-CoA, that's the acetyl group with the stabilizer, then it's ready to be sent into the citric acid cycle. Now remember, the whole point is to convert the energy from high value bonds, those are carbon-carbon and carbon-hydrogen bonds, into low energy bonds and capture the energy from that transfer. So all we're really doing is taking the remaining high energy bonds here, those are carbon-hydrogen and carbon-carbon bonds that are highlighted, and what we're going to do is we're just going to start breaking those apart and replacing them with carbon-oxygen or oxygen-hydrogen bonds, which are low value. and that's going to yield energy. Now, in the citric acid cycle, this happens via a variety of chemical reactions, and again, this is known as a pathway. The cyclical part of it is going to be ignored for now. Please note that we're going to transfer most of that energy into our coenzymes or our electron shuttles, known as NAD plus or FAD. They're going to be reduced into NADH and FADH2. So if you have a good understanding of the goal of the citric acid cycle, which is to finish breaking off the high energy nonpolar bonds and transferring that energy largely into NADH and FADH2, let's just do some bookkeeping. We don't need to get into the nitty-gritty of what the chemical reactions are. What we're really interested in is what is the outcome. So let's go back up to that shuttling step. So remember that when you shuttle pyruvate into the matrix, pyruvate is going to shed a carbon as CO2, and it's going to transfer hydrogen to become NADH. So what are you going to get out of the citric acid cycle proper? Now you're welcome to Google an image of the citric acid cycle and confirm this for yourselves, or you can just believe me. But here is the yield from one turn of this cycle. We're going to get 3 NADHs, we're going to get 1 FADH2, we're also going to get 1 ATP straight up. And then finally, we had 2 carbons in our acetyl group, and those carbons are going to get converted into CO2 along the way. So we'll get 2 CO2s. And remember, those yields are all going to be doubled because there are actually 2 pyruvates that are going to be shuttled in per glucose. All right, let's synthesize everything together that we've covered so far and do some bookkeeping of glycolysis and the citric acid cycle. So go ahead and copy this table down. We're going to do the starting reactant, the ending product, and what did we get out of that stage of aerobic respiration. So for glycolysis, we are going to start with a glucose molecule, and we are going to end with 2 pyruvate molecules, whereas the shuttle step and the citric acid step start with 2 pyruvates and end with 6 carbon dioxides. So just really quickly, you can see there that there are 3 CO2 circled, and then everything is doubled, so there's 6. Okay, what did we get out of this? Well, in glycolysis, we get 2 NADH and 2 ATP. We already covered that. Now, what about the citric acid cycle and the shuttle step? I want you guys to try this on your own. So what I'm going to do is just leave blank lines there, and then I'm going to take you back up to that picture, and I want you guys to count. So count everything up and then remember to double it, and you'll have your answer. Okay, this lecture is going to continue in the next video.